Welcome to the video on dissolution testing and method development of FAQs. Dissolution testing and method development is of prime importance in pharmaceutical field, and it is the compulsory topic for the questions in the interview. I welcome all of you to the video, and I hope after watching this video, after understanding this topic, you will be able to answer all the questions regarding the dissolution testing and method development. Dissolution topic is very crucial to understand, as dissolution testing is a in vitro tool or in vitro test to study the in vivo behavior of the formulations, and all the market formulations like Europe market, US market, Brazil market, ROW emerging markets, and all the markets including domestic as well. The regulatory authorities for these markets are always asking for the dissolution method development and testing. Many queries arise onto this topic, and that's why this is a favorite topic for the interviewers. So let's start with the question and answers quickly. What is dissolution testing? Why it is important in pharmaceutical development? Dissolution testing measures the rate and extent of the drug release from the solid dosage form. Also, for some of the liquid dosage form like suspensions, assessing bioavailability and ensuring batch-to-batch consistency. Dissolution is also a test to release the batch into the market. What are main objectives of dissolution method development? Objectives are evaluating the drug release, quality control, guiding formulation development, and supporting regulatory requirements of different countries. How does dissolution testing correlate with in vivo bioavailability? There is a term called IVIVC, so in vivo correlation. Dissolution is used to predict how the drug will dissolve and how the drug will be absorbed at what rate into the body. What are the key stages of dissolution method development for oral formulations or oral solid dosage forms? Key stages involve method selection, medium and apparatus choice, optimization, and validation. After this, the method is transferred to the QC lab. How does dissolution testing support quality control in manufacturing? Quality control. It ensures uniform drug release profiles across batches, impacting product safety and efficacy. What regulatory guidelines are relevant for dissolution method development? The guidelines like USP, USFDA, EMA, and ICH are focusing on the standardized approaches. How do you select appropriate dissolution medium? It is very important question. Selecting the medium depends upon the drug solubility and physiological relevance, like pH range of 1 to 7.5. Also, it depends on the BCS class of the molecule and the stability in the dissolution medium. How does pH of the dissolution medium impact drug dissolution? So, pH impacts ionizable drug solubility, and that's why dissolution. What is the role of temperature in dissolution testing? So typically 37 degrees Celsius temperature of the media is used to simulate body conditions, ensuring reproducibility and relevance. How is the agitation speed determined during method development? So generally in method development, dissolution is done for different RPM for basket and pedals, as agitation speed influences the hydrodynamic conditions simulating the GI environment. So based on to the experimentation, the RPM is finalized. What types of dissolution apparatus are commonly used for oral solid formulations or oral solid dosage forms? So USP1 basket, USP2 paddle for tablets and capsules, USP3 and USP4 for specialized formulations are used. How do USP apparatus 1 and 2 differ in their use for dissolution testing? The USP basket apparatus or USP1 apparatus is preferred for floating tablets, and paddle USP type 2 for with sinkers for the regular tablets is used. USP apparatus 2 is also used for some of the capsule formulations with sinkers float in the media. How do you calibrate a dissolution apparatus and why it is important? So calibration ensures equipment operates within specifications affecting test validity. What criteria should be considered when selecting a dissolution apparatus? Selection criteria. It depends on the dosage form properties and dissolution behaviors. What is the significance of sink condition in dissolution testing? See, sink condition is required to ensure the drug remains in solution, avoiding saturation and supersaturation and enabling the accurate dissolution measurement. So this is the answer for sink condition. Sink condition means the dissolution medium and volume is able to dissolve at least 3 times of the strength of the formulation. For example, if you are working on to 500 mg tablet, then the dissolution medium and volume should be able to dissolve 1500 mg of that API. How can paddle to tablet distance affect dissolution results? Paddle to tablet distance affects hydrodynamics, and that's why it affects the dissolution, because the tablets may get float. What is the impact of degassing the dissolution medium? Degassing prevents bubble from interfering with the dissolution rate, particularly with the USB apparatus 2. How do vessel size and shape affect dissolution outcomes? Vessel shape and size, you can say it as dissolution bowl shape and size, influences fluid flow patterns and dissolution, requiring standardization. What are the common sources of errors in dissolution testing? So sources are inconsistent paddle speeds, improper temperature and sampling errors. Many common errors may be there, like human errors, equipment errors and operating errors. How often should dissolution equipment to be qualified? So at least 1 year is the frequency. Calibration frequency at least once annually or after significant repairs to maintain the accuracy. How do you determine the appropriate dissolution profile of a drug product? Dissolution profile is determined by formulation design, intended drug release and desired bioavailability. What are the steps to develop discriminatory dissolution method? So we can develop it by formulation changes which affect the drug release. How do polymorphic form of the drug impact dissolution behaviors? Polymorphs differ in the intrinsic solubility and dissolution rates. That's why it affects the dissolution. Different polymorphs have different unique solubility and dissolution rates. What role does particle size play in dissolution rates? So smaller particles have increased surface area, and that's why they show faster drug release, while the coarser material have less surface area and it is slow in the drug release or dissolution rate. How does formulation type IR/ER influences dissolution method requirements? So IR formulations dissolve rapidly while the ER require controlled release. ER formulations are also known as sustained release, prolonged release or delayed release type of formulations. All these categories are under the common term of modified release. What impact do excipient have on dissolution method development? So excipient can impact drug release by altering dissolution medium properties or interacting with the drug. How do you ensure that dissolution method is robust and reproducible? So this question is very important for the analytical front. Robust and reproducible methods require optimization to ensure minor changes don't affect the results. So this is the best answer for this. Coming to the next question, what statistical tools can be used to optimize dissolution method parameters? Many statistical softwares are there, like DOE, design of experiments, helps to optimize parameters like pH, temperature and the agitation speed. Also for USB3 apparatus, you can use dips per minute as a speed. How do you validate dissolution method? So validation includes validating the accuracy, precision, specificity, linearity and robustness for regulatory standards and compliance. What are the acceptance criteria for dissolution method validation? So generally a consistent reproducible release profile meeting the QC standard is the criteria. How do you choose appropriate sampling times in dissolution testing? So it is chosen to capture the drug release profile based on formulation type. For example, for IR formulations, 15 minutes, 30 minutes, 45 minutes and 60 minutes is the time points. While for sustained release, 1 hour, 2 hour, 3 hour and later points are based on to the formulation like up to 12 hour or up to 16 hour or up to 24 hours. What is the purpose of performing sink and non-sink condition studies? So non-sink conditions are sometimes required for biorelevant testing, and sink condition ensures reliable dissolution. If you justify the dissolution discriminatory power, you can also use non-sink condition for some of the molecules. How do you handle drugs with poor water solubility in dissolution method development? So BCS class 2 and 4 drugs are having generally the poor solubility, and these require media with surfactant or adjusted pH to improve the solubility. What approaches can be used to improve the discriminatory power of dissolution test? So improving the discriminatory power involves using different pH levels, surfactant or varied agitation. How do you handle or how do hygroscopic properties of drugs affect the dissolution testing? So hygroscopic drugs are those drugs which absorb moisture or water from the environment, and thus these may have effect onto the dissolution consistency and stability. So proper handling is required for these type of formulations. Also, light sensitive drugs are handled by having the amber colored bowls. What strategies are used for developing dissolution method for poorly soluble drugs? Generally, surfactants are used to develop method for low soluble drugs, or co-solvents are used to improve the solubility. But co-solvents like organic solvents are required to be restricted. How can formulation modifications impact dissolution outcomes? Formulation modifications may involve changing excipient or using solubility enhancing techniques like solid dispersion, use of surfactants, and others. What is the role of surfactant in dissolution media for low solubility drugs? So surfactants are generally used like SLS is there, Triton X, Samtrid is there, P80 is there. These improve dissolution of lipophilic drugs by reducing the surface tension and improving the weighting. How does polymorphic conversion during dissolution affect method reliability? Polymorphic conversion and polymorphs can change dissolution rate requiring monitoring during testing. So if polymorphic conversion is more, then it will be reflected in the dissolution. How do storage conditions affect the stability of dissolution medium? If you store the dissolution medium for longer time, it may cause medium degradation or evaporation and affects the test results. What are the common regulatory requirements for dissolution testing in oral formulations? So these include meeting pharmacopoeial standards and ensuring the IVIVC. Also, the discriminatory power is required to be proved. How the FDA guidance on dissolution apply to control release formulations? So it requires extended dissolution testing and potentially in the multiple media, that is known as multimedia testing. What role does the biopharmaceutical classification, that is BCS, play in dissolution testing? So generally BCS class 1 and 3 drugs are eligible for biowaivers, while the BCS 2 and 4 are not eligible. How is the dissolution method transferred between laboratories? So method transfer involves comparative studies and adjustments to ensure reproducibility across labs and across the sites. How do regulatory bodies assess the robustness of the dissolution method? So regulatory bodies check the consistency under various conditions or varied conditions, and that's why check the robustness. What are the common pitfalls in submitting dissolution methods to regulatory agencies? So common problems are like insufficient discriminatory power or inadequate validation data. Also, there should be a study on solubility, equilibrium solubility and BCS solubility to be submitted in the dissolution method development. What is the significance of IVIVC in dissolution testing or regulatory approval? IVIVC links dissolution and bioavailability, reducing the need for extensive in vivo studies. How can dissolution profile be used to support generic drug approvals? So dissolution profile similarity is required to be demonstrated to have justification for equivalence to the reference products. How do we approach method adjustment for scale up and post approval changes? So testing verifies that changes do not affect the drug release profile, and thereby we can do the approach for method adjustments. What documents or what documentation is essential for regulatory submission of dissolution method? So it includes SOPs, method validation data, and testing results for the regulatory audits. So these are the common topics for dissolution testing and method development for interviews. I have made many videos on dissolution testing. So please watch those videos and you will have good information for these topics. I hope after watching those dissolution videos, you will be a very well versed, knowledgeable person in this topic and you will be able to answer all the dissolution related questions. Whether you are a analytical person or whether you are a formulation R&D person, QC person, regulatory affairs person, or pharmaceutical professional. So this was the topic. This was the information for frequently asked questions. Also prepare the list of questions on your own and try to find out the best answers. Thank you for watching the video and thank you for visiting the Pharma Learning in Depth channel.